I think that the first thing which is important is that if you are going to build your own facility, prior to not the design, but in parallel to the design of the facility, you need to start preparing risk assessment about cross-contamination, because that will tell you how to build the facility. For instance, there are facilities that they have areas in which you can work both with mammalian cells and with microbial cells. That is not allowed. That is huge risk of cross-contamination.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brulman, and this is part 2 with regulatory expert Rivka Zaibel, who is the founder and president of ADRES. Today, we tackle preparing for regulatory meetings, the dangers of cross-contamination, and common misconceptions that might be derailing your CMC development strategy. Let's explore powerful strategies to regulatory compliance and CMC development. Let's continue our conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now, I have had throughout my career some interesting conversations with people from quality assurance and regulatory affairs. So, we are in a highly regulated industry, no doubt about that. But sometimes there is also a gray zone. And finally, the question sometimes comes up, what is good enough? In particular, when you are a smaller company with limited resources, what would you tell someone in that space? What is good enough? And let's make it very practical. What are the key success factors and maybe some common pitfalls as you're preparing your regulatory filing?
Okay, so it's not a yes-no question, or I can give you a recipe of what you can do, but it's more a risk-based approach to as it is for everything. I think that one of the advantages that I have being 36 years in the industry is that I can evaluate what can be accepted by the regulatory authority and what will not be accepted no matter what. So anything that has to do with safety, you don't try to cut the corners there. You need to do everything that is needed. For instance, the issue that I mentioned before, the viral clearance studies, it's not something that you can negotiate. You need to do that. You need to find the way to do the minimum that is needed at the stage of development so that you don't really waste time and money on things that are not really required. But one of the things that my clients know that is that when I can suggest solutions that are less demanding, then I will do so. But when I say that something is needed because otherwise I cannot represent you to the regulatory authorities, that's because that's the case. It's not, I know that there are other consultants that will insist on everything, and then it is difficult to work in that world. But the idea is that you need to risk assess each one of your decisions and think what might be the outcome of not fulfilling the requirements. Not only in the discussion with the regulator, but also in the development of your product. Sometimes when you cut corners, you don't get to the product that you wanted to get because there are things that you couldn't actually think about that and then you couldn't prevent them from happening. So it is a constant risk assessment.
And what are some pitfalls you have observed throughout your career?
So one of the things that I see actually today, even today, is that there are some aspects, for instance, of facility cleaning and disinfecting that are not being taken into consideration during the initial phases. For instance, you need to validate the efficacy of disinfectants that you use in order to disinfect the equipment, the facility, the areas in which you work. Not all of the companies actually conduct those studies in early phases. Now, that is a requirement both by the FDA and the European authorities. Annex 1, that is a document that everyone in the industry, mainly those that are working in aseptic processing, they need to work according, and there are a lot of discussions about that, is very clear about the fact that you need to do the disinfection studies, the efficacy of the disinfectants in your facility with surfaces in which you work and you are going to use those disinfectants, and you cannot rely on what the supplier gives you. So there are still companies that they rely on what the supplier provide them. And at the end of the day, if they are going to be subject to a regulatory inspection, it will not be acceptable. There are other areas, for instance, that I see a lot of operators which are not trained enough to work in those clean environments. And this is an area that companies need to invest more.
How do you go about to avoid cross-contamination? Because you mentioned disinfection and the training, and I mean, it can get very quickly very complicated with different products.
That's right. And so actually cross-contamination is a subject for a seminar, not for a 20 minutes discussion.
Yeah, absolutely. We should do a second episode about it.
Yeah, we can do that. So I think that the first thing which is important is that if you are going to build your own facility, prior to not the design, but in parallel to the design of the facility, you need to start preparing risk assessment about cross contamination, because that will tell you how to build the facility. For instance, there are facilities that they have areas in which you can work both with mammalian cells and with microbial cells. That is not allowed. That is huge risk of cross contamination. And if you want to manufacture both kinds of products, then you need to have totally segregated areas for mammalian cells based products and to microbial cells based products. So that is something that you need to actually incorporate into the design of your facility. It's not something that you can leave for the future. I've seen facilities that after inspection, they had to rebuild the facility or redesign the manufacturing areas because of that. The other aspect that you need to be very concerned and understand it is the issue of cleaning validation of the equipment and the facility. Also, that is something that needs to be incorporated into your risk assessment. What is your more risk product that you intend to manufacture in your facility? And based on that, design all the activities and the procedures. The training of employees, that is critical for cross contamination. Sometimes you actually don't understand how something is contaminated, and when you look back, you realize that there is one operator that constantly has out of specs results in his environmental monitoring testing. Or you observed the operators working in the clean area and that they touch areas that they shouldn't touch. They don't disinfect the hands often enough. Those are little things that you have, if you are not aware of them and you don't treat them and you don't resolve them, then you will have a huge cross contamination issue. And you also ask in your one of your questions about what the outcome of that can be, and there are facilities that were closed for months or more than that because of that, because they didn't realize that they have a contamination issue, and then when it came up, it was something that actually forced them to close the facility, to change the equipment. There was one case of drug shortage because of that, which seems to be something that if you have detected early enough in the process, it could have been resolved.
Yeah, and it comes down to a proper risk assessment again.
That's correct.
To know your process, also what you said in the beginning. Now, Rivka, I just want to circle back to something else you mentioned in the beginning of our discussion. You mentioned that the regulatory landscape is evolving. We have new products. We have now cell and gene therapies, DNA products, RNA products. I want to just touch upon one aspect, which is the cell therapy space, because now we are moving away from the process is the product. Actually, we are now using the cells that are the product. So how does now the regulatory aspect change there?
Okay, so first of all, the requirements are that for cellular therapy product, the aseptic process starts at the very beginning. Every step in the process is in aseptic conditions because you cannot sterile filter the cells and you cannot sterilize them at the end because then you will destroy the product. So the challenge of manufacturing them is really very high. But today there are closed systems that were developed to serve that industry. So more and more sophisticated process can be performed within a closed system. Still, you need to be very trained to operate those systems. You need to be aware of stages in the process that you may not be able to use a closed system, and then what are the conditions by which you continue the process so that you don't contaminate that. It is a challenge area, but what I've seen in different facilities is that there are solutions. And if you understand good enough your process to see where are the areas that are more risky, then you find the right solution to address them. And here also the training of the operators is very, very critical.
Now, we have done our homework. We have now done a proper risk assessment. We know our process. We know what are the different steps we have to do in order to have a proper filing. Now, the time comes now that we are now ready for the first regulatory meeting, and you have been part of many meetings in your career. What advice would you give us here to prepare properly and then also have a successful meeting with a regulatory agency?
Okay, so first of all, I would say that I wouldn't wait until you have proper risk assessment and everything else to go to the first meeting. I go earlier than that. I go with my plans. For if I want to have a multi-product facility, then I would go to the FDA to consult with their GMP team about the design of the facility and to have their agreement that the way that I design the facility is according to the regulation and their requirements. And also by doing that, you get also advice about things that you maybe didn't think about. But if I am in a more advanced step, and I have already an idea of what is the process that I'm going to use. From a CMC perspective, I would go to the FDA prior to the manufacturing of the first GMP batch to have their feedback about the process, the specifications, the release test that I intend to do, the validation of the most critical methods. For instance, the safety methods need to be validated prior to phase 1 for the GMP material. And that is in general what the scope of the meeting would be. And I would think about all the questions that I have. And I go there and ask whatever bothers me, whatever I'm not sure about. I don't think it's wrong to ask questions. It's the only way that you will know whether they agree with you or not. A lot of companies fail asking questions during the development, and they submit that to the IND, and they think that because they submit the data and their concept to the IND, then and the FDA didn't have any comment about that, then it is okay. And then companies submit their BLA and they get a complete response letter in which some of the things that they have submitted throughout the years are there as things that the FDA doesn't agree with it, and they say, but I told them about that. It's not about telling them, it's about asking. If there is something that you are doing that is not exactly according to the regulation, according to the guideline, if you are not sure whether it is accepted or not, you need to ask. And they will tell you it is acceptable, it is not acceptable. If you want to go this way, then I suggest that you do some additional things. So if you don't ask the question, it doesn't mean that they agree with everything that you have submitted. And I've seen companies really getting the complete response letter and saying, but we submitted all of that years ago. How come they don't agree with that? So my message is don't be afraid to ask a question.
That's great. Thank you so much, Rivka, because meeting a regulatory agency can be scary. But what you're saying, it's okay to ask questions, and it's also okay at this stage not to know everything, because it's meant to be a dialogue, right, with the regulatory agency.
And also don't be afraid of sometimes challenging them. Sometimes it works.
That's wonderful. We have covered so much ground today, Rivka. With all that we have discussed, what is the most important takeaway you want our listeners to walk away with?
To understand the product, to think about what will be the final product that I want to bring, and then plan back how to get there. Not only in terms of the process, but also all the supporting aspects that are needed. I think that's the most important thing.
Thank you so much, Rivka, for sharing this wealth of information, this wealth of expertise and experience. It's very useful to our community. Where can people get a hold of you?
So first of all, we have a website, www.adres.bio, and also my personal email. If you wish to publish that, that's fine.
Okay, great. I will leave this information in the show notes, so please reach out to Rivka. And Rivka, once again, thank you so much for your time and for sharing all this insightful information. It was a huge pleasure to have you on the show today.
Thank you very much. I enjoyed that very much.
And there you have it. Rivka's practical roadmap for navigating the complex world of biologics development gives you tools to overcome key challenges in bringing therapies to patients. I'd like to offer you a free assessment to evaluate and refine your CMC development strategy with precision. If you're feeling overwhelmed by regulatory complexities, book a free call with me through the link in the show notes. Join us next time as we continue to simplify bioprocessing. This is David Brulman reminding you that science may give you headaches, but biologics drug development should not. Alright, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.